Hello, class. How are you doing? Chapter 18. It's a little bit, I don't know if I finish it in 1 hour. I doubt that. I think this chapter will take a little bit more than an hour. But the bulk and bold, the big nuts and bolts of this chapter pretty much tell you the whole chapter in one sentence. And then we have to go over that sentence for a while. It is just like a switch, a light switch that is on, and then you come and turn it off. A switch that is on all the time. And the light switch that you go into a room and you turn it on and you turn it off. So that's what this chapter is all about. I'm going to talk about how a gene can be turned off and turned on. Some genes are on all the time, so they need to be turned off. And some genes are not on at all the time. They're not on at all, but there are some factors and molecules can turn them on. And then of course they have to be turned off again. So I hope I made some sense. It is just look at this, nothing on, off, and now we are on. Okay, so that's the best analogy I can make for you guys. Let's go ahead and see what the chapter has all about in here. I have to do some reading and talk about that. So anyhow, bacteria often respond. This chapter is not about bacteria all the time. There are some eukaryotics, but we still are using E. coli to get a lot of information. Most of the discoveries have been made from studies that have been done on E. coli, the bacteria E. coli. So anyhow, bacteria often respond to environment change by regulating transcription, and natural selection has favored bacteria that produce only the products needed by that cell. A cell can regulate the production of enzymes by feedback inhibition or by gene regulations. So that's pretty much the bulk of the chapter is about by feedback, and then if there's too much of a molecule inside of the cell is turned off, and if there's not enough, then it has to be turned on. One mechanism of control of gene expression is bacteria is operon model. So there are a few mechanisms, but the one that we are going to focus on this chapter is operon model. Okay, so the mechanisms of the gene control. So that would be a nice multiple choice question. Control of gene expression. You know, if you know about gene expression, making a blue eyes. You know, it's making a blonde hair, making a black hair. Those are gene expressions. Okay. So let's take an example that, for example, this is a precursor to the amino acid tryptophan. And then what happens, these are the genes involved, the DNA, TRPA and TRPD, and TRPC and TRPB and TRPA. And all of these 1, 2, 3, 4, 5 genes on the DNA, regulation of gene expression, they are involved to make 3 enzymes. So DNA make enzyme 1 and C make enzyme 2. You don't have to memorize this. I'll let you know what you have to know. And BNA enzyme 3. So enzyme 1 is going to take the precursor into making this molecule, which is not the tryptophan yet, the original tryptophan. It's not that one yet. So, and then this precursor become this molecule with the help of that enzyme, enzyme 2 and enzyme 3, which gene A and B are responsible to turn to make this enzyme, enzyme 3, take that precursor and make tryptophan. Okay, so what happens, the tryptophan excess tryptophan in the cell, it goes, and as you know, tryptophan is an enzyme, is an amino acid. Oh, come on. Is an amino. I didn't have coffee this morning, so that's why. It goes back and turn off. It tells that enzyme, that gene, right here, to stop. And also, it would excess tryptophan goes into the enzyme, and we studied that at the chapter 4, 5, 6. That enzyme, it stops. It's a allosteric enzyme, so it can stop that enzyme from converting this precursor to this molecule. Okay, I hope I made some sense. So, having said that, let's move on to the next one. Operon system. So what is an operon system? A cluster of functionally related genes, and you saw that, A, B, C, D, E, in this case. A cluster of functionally related genes can be coordinately controlled by a single on and off switch, the tryptophan. Okay, and then the switch is a segment of the DNA called an operator, and you will see that in the next picture. So the switch is actually an operator, is on a DNA. I'm going to use that as a DNA. A segment of that DNA right here is the operator. Okay, an operon system is the entire stretch of DNA that includes the operator, the promoter, the genes that they control, the A, B, C, D, E. Let's look at the picture. The operon can be switched off by a protein repressor. Okay, you will see that. Now, I guess not makes a transparency and a few one down. And the repressor prevents gene transcription by binding to the operator and blocking RNA polymerase. Okay, so RNA polymerase, as you know, is responsible for protein synthesis, and then for transcription, the beginning of it, the RNA polymerase is responsible for transcription, make the mRNA. You remember that from last chapter, and then mRNA goes to the cytoplasm and makes DNA, makes protein molecule, makes the protein molecule. The repressor is the product of separate regulatory genes. Okay, anyhow, the repressor can be in an active or inactive form depending on the presence of the other molecules. Yes, a corepressor is a molecule that cooperates with the repressor protein to switch an operon off. Okay, an example, for example, E. coli can synthesize the amino acid tryptophan, but it has insufficient tryptophan, which you saw that a little bit. By default, the tryptophan operon is on, so tryptophan is on all the time by default. Do you remember I talked about there are two switches, the bulk of chapter. So tryptophan always, the cell always tell the precursors to tryptophan. In E. coli, again, we're talking about E. coli. And the gene for tryptophan synthesis is transcribed. Okay. When tryptophan is present, when there is tryptophan present, it binds to tryptophan repressor protein, which turns operon off. So repressor turns the operon off. It's always on. Excess tryptophan, turn it off. Okay. The repressor is active only in the presence of co-repressor tryptophan, thus the tryptophan operon, TRP, they call it TRP. Operon is turned off, repressed if tryptophan level is high, which I already talked about that, right here. Okay, so here is the regulatory gene, DNA promoter right here. Let's start from here. DNA promoter and the regulatory gene, they're all right here. And then, of course, you know the RNA polymerase right here. That's an RNA polymerase. And that segment of the DNA, look at it, the promoter. Okay, this whole entire thing is operon. You can see that. Now the operon includes tryptophan, the segment, the gene, tryptophan E, D, C, B, A. So it includes those, the operon. And operon includes the promoter. Okay, I'm just talking about DNA so far. Hang on. And then, of course, the mRNA is synthesized. You can see that. And based on the mRNA, enzymes A, B, C, D, E are made up. Okay. So having said that, this gene is on all the time. Okay. And this is an example of a negative control. So I said that. Tryptophan absent, there is not enough tryptophan inside of bacteria. So tryptophan absent, this is on all the time and it starts making tryptophan based on, from a precursor, which you saw it, okay? Now on the bottom, you can see the tryptophan is present and this is a repressor protein molecule. That repressor protein molecule, tryptophan attaches to it and it goes right up here and sits on that operator. The operator right here, it sits in there and it prevents RNA polymerase from doing its job, which is transcription, making the mRNA right here, making the mRNA right there. And then they put this diagram for you here in this diagram that you know what we are talking about. We are talking about this PowerPoint is focusing on the DNA right here and here and here and here. And they're all in one gene. Okay, it's all of one DNA, I'm sorry. Okay, I hope I'm making some sense. Here is operon on when there is not enough tryptophan in the cell and the repressor is inactive. But when there is a lot of tryptophan inside of the cell, what will happen? It goes and sits. It attaches to that repressor and the repressor become active and it sits on that segment on the operator. They didn't mention it right here. Operator. It sits on the operator and then, of course, RNA polymerase right here. RNA polymerase cannot go downstream. You remember downstream and upstream? This is downstream. This is upstream. So be familiar with those terminologies a little bit. You cannot go downstream. Okay, responsible, maybe I should go get coffee. Repressible and inducible operons, two types of negative gene regulations. A repressible operon is one that is usually on. The light of the room is on constantly. That is responsible. That is called repressible operon. It's an operon can be repressed. Is usually on. Binding of a repressor to the operator shuts off transcription, light switch on all the time. Okay, the tryptophan is an repressible operon. The example you saw. The inducible operon, it means it's usually off. This light switch is off of the room. Okay, a molecule called the inducer inactivates the repressor and turns on the transcription. The light switch off all the time. This is a case of the light switch is off, the gene is off all the time. The gene is off all the time right here. But what happens, there is a molecule called inducer, and then it turns the switch on in inducible, and then we'll talk about that here. The lac operon, okay, is an inducible. I did not give you an example of inducible. What you saw, it was a repressible. So the lac operon is an inducible operon and contains genes that codes for enzymes used in the hydrolysis and metabolism of lactose. And you know lactose is a disaccharide. We talked about it at the beginning of semester. It's a disaccharide which is made up of glucose and galactose. By itself, the lac repressor is active and switches a lac operon off. Okay. A molecule called an inducer inactivates the repressor to turn the lac operon on. The turn off switch on. Okay. Do we have a picture? Yeah. Is this the lac? Yes. Is this still example of a negative control? So the switch is off. Here is a repressor, is always here. And what happens, do I have, yes, operon off, okay, RNA polymerase cannot go downstream. That's a normally happens in the case of this type of gene. This is a different, look, this is a different type of gene. This is, for example, look at the diameters. I don't know, I wish I had a different one. See, they're different sizes. You can see that. So this is a lac gene for lactose, and this is a trypt gene. Okay, so this one, this end right here of the cord, it's off already. Now what will happen, so you have allolactose, it will attach to the inducer, inactive repressor, I'm sorry, to the repressor. So the repressor is off from operator, it goes off, and then what? RNA polymerase is going to continue going downstream and do its job, make these enzymes right here, and those enzymes will make lactose. Okay. So what this is, I should have brought it up, but that's okay. You guys are good. Inducible enzymes usually function in catabolic pathways. As at the beginning of semester, you know what catabolic pathways means. Pathways, they are catabolism, it means breaking down. Anabolism, it means building up. So inducible enzymes usually involved in catabolic pathways, synthesis induced by a chemical agent. Repressible enzymes usually function in anabolic, putting molecules together. Synthesis in repressor by high levels of the end product. Regulation of tryptophan and lac operons involves negative control of genes because operons are switched off by the active form of repressor genes. I have to keep on doing this. Okay. Positive gene regulations. Let's, you study 2 of the negative gene regulations. Let's do one positive one. So, you know, you get a feeling for that one too. It's Chapt, usually referred as Chapt. Some operons are also stimulatory proteins such as catabolic activator proteins, CAP, or an activator of transcription. When glucose, a preferred food for E. coli, as I said, is the bacteria, is scarce in the cell, then Chapt is activated by binding, that is, and activated Chapt, catabolic activator protein, that's what it is, attaches to promoter You know what that is on the DNA. Okay, sorry, we got we got disconnected. I guess my internet was unstable for a moment. So now we are back into business. Oh, come on. So activated Chapt attaches to promoter of the lac operon and increase the affinity of RNA polymerase, thus accelerating transcription. When glucose level increases, Chapt detaches from the lac operon and transcription returns to a normal rate. Okay. Chapt helps regulates other operons that encode enzymes used in catabolic pathways. Here is an example of a positive control. So it accelerates the enzyme transcription and accelerates transcription. You can see the promoter on top, and then you can see cyclic AMP attaches to catabolic activator protein, the Chapt, right here. Cyclic AMP attaches to Chapt, and it goes in here, and then of course it promotes the cyclic RNA polymerase and binds to transcribes, and then it moves on. Okay, and then allolactose attaches to the repressor, so it moves on. But when there is not enough cyclic AMP, catabolic activator protein is here, and of course do I have one? And I'll talk about this one right here. So lactose present in a cell, glucose present, cyclic AMP level is low, little lac mRNA synthesizes occurs. Okay, so you have enough lactose and glucose as a backup. Okay, differential gene expression. Almost all the cells in an organisms are genetically identical. Differences between the cell types results from the cells. What this very first sentence is saying, that the cells in my eyes and the cells in my skin are same, this genetically. Okay, the cells in my liver and the cells in my hair follicle are same, gene-wise. Okay, differences between cell types results from differential gene expression. The cell in my hair makes a different protein molecules. The cells in my eyes make a different protein molecules, but genetically they are same. I hope I'm making some sense. Differential gene expression, the expression of the different genes by cells with the same genome. Okay, so abnormalities in gene expression can lead to diseases, including cancer, which we will talk about it. Gene expression is regulated at many stages. So that's another thing, yes, we need to talk about. Here is the regulations, it's a diagram that shows the regulations of a gene. Eventually, what happens, each box indicate a type of regulations. And then, of course, it says here, DNA regulations in blue box, right here, RNA regulations in red and orange. These are all RNA regulations. And then protein regulations in blue boxes. And then we will talk about it. So all the way from transcription, which is right here, all the way down to translation and modifications, there is control along these steps. Okay, so signals goes to DNA. Here is a chromatin right here. Signals go to chromatin and chromatin modifications, DNA unpacking. That's the next thing I'm going to talk about. DNA gene available, and then transcription, translations, will occur next. So let's talk about that. These are diagrams. Please study them. Oh, you know, what is he saying?
A cell can get rid of abnormal or damaged proteins and limit the lifetime of functional proteins by means of selective degradation. The cell marks the protein for destruction by attaching small ubiquitin proteins. Giant protein complexes called proteasomes recognize ubiquitin and break down the tag protein.
Very good. Okay, so ubiquitin has to attach to the protein that has to be degraded, and then another one.
Often, polypeptides must be modified in some way to produce functional proteins. For example, a polypeptide is cleaved in 2 or more polypeptide chains. Other molecules might be attached to the protein, or the protein might be phosphorylated. Some proteins need to be transported to specific destinations.
Okay, yeah, these are all the things we studied at the beginning of semester.
Translation of specific mRNAs may also be blocked by regulatory proteins that bind to sequences within the untranslated region at the 5' end of the mRNA molecule, modulating the amount of protein produced.
Okay, that's another regulatory mechanism. So regulation of chromatin structures, let's talk about that. The structural organization of chromatin helps regulate gene expression in several ways. Chromatin, you know, if this is DNA, we talked about it, right? Here is all bundled up, right? Here it is chromatin now, right? You all know that. This is a chromatin right there. Okay, so genes within a highly packed heterochromatin are usually not expressed. If there's a gene way in here, that usually does not get transcribed and translated, okay? Chemical modification to histone, you know what histone are? They are protein molecules that DNA wraps around them twice. So my fist is histone, and this is the DNA wrap around it twice. So, and then extend to another histone right here, twice. Oh, yeah, this is twice. That's what it is. Okay, my fist are histone, protein molecule, DNA wraps around them, and then we'll talk about it. Chemical modification of histone and DNA chromatin influences both chromatin structures and gene expression. Yes, right here. Very, very, very important diagram that you guys should know. PowerPoint slides. In histone, acetylation, so this is the structure of acetyl, right here, acetyl tail. The histone in acetyl group attaches to the lysine. Lysine was what? You guys remember? An amino acid, okay? And this attaches to lysine, so it loosen up. So this is DNA, it's condensed. So acetylation, it loosen it up a little bit. Okay, this is condensed, chromatin is condensed. This acetylation loosen it up. Acetylation promotes the initiation of transcription, the beginning of transcription. And the addition of methyl group, methylation, can condense chromatin. So if this is chromatin, and then you want to make it opposite of acetylation is methylation, right here. Okay, so what else? The addition of phosphate groups, phosphorylation next to methylation, amino acids, it loosen up. So combination of phosphorylation, we talked about at the beginning, adding a phosphorus group, and then methylation together, it loosen it up. Okay, so here they are. At the end, these are lysine attached here. These are the histone protein molecules. DNA wraps around, and then what happens? Acetylation makes the loosen up. This is unacylated, acetylated, and then this is histone, and then this is acylated histone. So again, chromatin, so we studied at what level gene regulation can occur in the chromatin level. Epigenetics, inheritance, I do have a website for you guys in here. I highly recommend, please go and watch it. It's in YouTube, I hope it's still there. But epigenetic is, although the chromatin modification just discussed do not alter DNA sequences, the DNA does not change. They may be passed to future generations of the cells. The inheritance of traits transmitted by mechanism not directly involved the nucleotide sequences is called epigenetic inheritance. Example, identical twins, one is normal and the other one has schizophrenia. So that would be an example of epigenetic inheritance that we studied, that we talked about it. Organizations of typical eukaryotic genes associated with most eukaryotic genes are multiple control elements, segments of non-coding DNA that serve as binding sites for transcription factor that helps regulate transcription, yes, and control elements and transcription factors they bind are critical to the precise regulation of gene expression in different cell types. That's what we are going to talk about next. Typical organizations of eukaryotic genes. So that's what this is. You have exons and introns. We talked about it, the promoter, we already talked about that. Exons, they exit. These two are going to be combined together. Introns will remain behind, and exons, they all get together. And then, of course, at the beginning, you have a, they call it untranslated region, UTR, right here, the cap. You know, we talked about this, I think, and then polytail, so the cell knows it's the end of it. And then you have another UTR at the end. The stop codon are right here. Start codon is right here. AUG, start codon. You remember that? And then this is the coding segments. So that's eukaryote. That's what a gene look like. They're talking about it. They're giving you another.
The lifespan of mRNA molecules helps determine how much of a protein is made.
That's right.
Nucleotide sequences that affect mRNA longevity are often found in the 3' untranslated region following the stop codon. Enzymes attack the poly-A tail first, and eventually the rest of the mRNA. Depending on its ability to resist this degradation, a eukaryotic mRNA molecule may last from hours to weeks. The longer an mRNA lasts, the more protein is made.
Okay. The role of transcription factors to initiate transcription, eukaryotic RNA polymerase requires the association of transcription factors, which we talked about those a little bit. General transcription factors are essentials for the transcription of all proteins codings. So we talked about that, the promoters and repressors and so on and so forth. In eukaryotic, high levels of transcription of particular genes depends on control elements interacting with specific transcription factors. And I will talk about those, the new transcription factors here in a minute. The difference between liver cells and the cells in the eye is coming up. Enhancers and specific transcription factors, proximal control elements are located close to the promoter, and distal control elements, groupings of which are called enhancers, may be far away from the gene or even located in an intron. The two activators, I'm not crazy about this, the two activators of proteins to promote transcription, the domains, DNA binding domain and activation domain, I think I should have deleted those, but an activator is a protein that binds to an enhancer and stimulates transcription of a gene. An activator have two domains, one that binds the DNA and a second one activates transcriptions. Bound activators facilitates a sequence of proteins for protein interactions that results in transcription of a given gene. Okay, so Now activators, we talk about this, and then group of mediators, proteins, and you know, that trompin, you know, we talked about this, the trompin effect. Right here, is it this one? Probably say
Most of the fine-tuning of gene expression occurs at the transcription level, making an RNA copy of a DNA sequence. Recall that to initiate transcription, an RNA polymerase must bind to the promoter region adjacent to a gene. This cannot occur without the help of specific protein transcription factors. Some transcription factors, called activators, bind to enhancer regions some distance from the gene. The DNA bends, bringing the activators closer to the gene, where the activators interact with other transcription factors and RNA polymerase to initiate transcription.
Right. Okay. So these are some of the things I think was from last chapter. Combinatorial control of gene activation. A particular combination of control elements can activate transcription only when the appropriate activated proteins are present, right here. That's what I was talking about, the difference between liver cells and lens cells. Lens cells in your eye and liver cells inside of your liver. Look at these molecules. What makes a liver cell different than, they have the same gene. They have the same gene. All of your body have the same gene, the same DNA sequences, the same nucleus, if you would. What makes it different between the cell of your eye and the cells of your liver? Right here, specific transcription factors are made in particular cells. These protein molecules are made in liver cells, and then the transcription of the albumin. Albumin is a protein molecule your liver makes, okay, is expressed because of these. These are the activators, if you would. Specific transcription factors are made in a particular cells. Okay, the crystalline, that's another protein molecule which is from your eye cells. So what happens, these are the activators are different. Available activators are different than the liver cells activators. So as a result of that, different portion of the cell is going to be transcribed and eventually translated. So crystalline is going to be. I hope I made some sense. Here they are. So comparing the two, and here is the liver cells, and here is the liver cells, and here is the crystalline gene is not activated in liver cells. They have it, but they're not activated. Why they're not activated? Because of these protein molecules. They can only, they will activate that portion of the cell which is responsible for albumin. Here is the lens cells right here. These activators will not have any effect on that segment of the DNA. They have effect on the crystalline end of the DNA. Okay, so let's talk about RNA a little bit now. The RNA processing in alternate RNA processing. Let's see how long I've been talking about this. Oh, I forgot to turn my, I've been talking about, I guess, for 20, 30 minutes. 20 minutes, I guess. So in alternative RNA splicing, different mRNA molecules are produced from same primary transcript depending on which RNA segments are treated as exons and which are treated as introns. Right here, you know, I'm sure you can study these and you already know about it. We talked about it a little bit, RNA processing. Oh, let me go back and play this one.
A cell can rapidly adjust to environmental changes by regulating several processes that happen after transcription. In RNA processing, a cap and tail are added, introns removed, and the remaining exons spliced together. Alternative splicing of RNA may create different mRNA molecules from the same primary RNA transcript.
Yeah. Okay. So Initiation of translation and mRNA degradation. The lifespan of mRNA molecule in the cytoplasm is a key determining protein synthesis. Eukaryotic mRNA is more long-lived than prokaryotic mRNA. Okay. Quiz question. Nucleotide example question. Nucleotide sequences that influence the lifespan of mRNA in eukaryotic resides in the untranslated regions of the 3' end of the molecule. Anyhow, after translation, various types of protein processing occurs. Protein processing and degradations, yes, that is also important. So what happens, the length of each protein function is regulated by selective degradation. That's the ubiquitin. Ubiquitin was described later. Ubiquitin comes and attaches to a protein molecule. That protein molecules goes to proteasomes, and proteasomes break it apart. I cannot break my finger apart to pieces, but you already saw the video. You know what I'm talking about. So recognize it, which recognize and destroy it. Let's see.
A cell can get rid of abnormal or damaged proteins and limit the lifetime of functional proteins by means of selective degradation. The cell marks the protein for destruction by attaching small ubiquitin proteins. Giant protein complexes called proteasomes recognize ubiquitin and break down the tagged protein.
Okay, you already seen that. Non-coding RNAs play multiple roles in controlling gene expressions. Only a small function of DNA codes for proteins, and that was surprising in 2003 when they figured out the sequence human genome, and only a small portion function of DNA codes for all of the proteins possible in human, and a very small fraction of the non-protein coding DNA consists of genes for RNA such as rRNA and tRNA. A significant amount of genome may be transcribed into non-coding RNAs. Non-coding RNAs, it means RNA that are not responsible for making any protein molecules. It's called non-coding. You remember the mRNA, the 3 codon? The 3 codon on mRNA is called a codon. That's what you're talking about. So it's for rest of PowerPoints, it refers as NC, non-coding, NC RNAs. Non-coding RNAs regulate gene expression at 2 points: mRNA transcriptions and chromatin configuration. Chromatin configuration means the change of the shape of the chromatin. So if you have a chromatin like this, if it changes its shape, okay, that is happening. We already talked about that in some detail. That is happening at the non-coding RNA, which makes sense. Effects on mRNA by microRNA, small RNA, and small interfering RNA. MicroRNA, miRNA, are small single-stranded RNA molecules that can bind to mRNA. Okay, so if you have an mRNA like this, miRNA binds to mRNA, and then these degrades the mRNA or block them its translations. It is estimated that expression of at least half of all human genes may be regulated by mRNA. Here it is, mRNA, microRNA protein complex, and here they are showing you how they can be degraded and so on and so forth. So at mRNA level, degradation control, we're talking about gene control at mRNA level. Small interfering RNA, siRNA, are similar to microRNA in size and functions, and the blocking of genes expression by siRNA, small interfering RNA, is called RNA interference or RNAi. So that is a good thing to know for quizzes exams. RNAi is used in the laboratory as a means of disabling genes and investigate their functions. They use that scientists, if they want to see the expression of other genes, they block some genes that do not want to see this protein molecule in the bacteria, for example, that it is making. So they block them with that RNAi. Chromatin remodeling by ncRNA, non-coding RNA. So some non-coding RNA act to bring about remodeling of chromatin structures in some yeast. RNAs reform heterochromatin at centromeres after chromosomes replications, heterochromatins condense, and then chromatin areas, not much of transcription. So if you want to block something not to make it, you condense it by adding methane, and then it's not. Here is a chromosome centromere system chromatids. You guys, I'll let you study this. Again, we talked about all different types of RNA. We talked about usually during a normal semester when it's in person. I do not talk about this either. I'll let you read it, and non-transcription and translation occur. So here, quickly, transposons. I remember when it came out, everybody talked about parasites, but it's interesting. It's an interesting concept. Small non-coding RNA called piwi-associated RNAs. So they are induced heterochromatin blocking the expression of parasitic DNA element in genome known as transposons. So the transposons, what is a transposons? A segment of DNA that is capable of inserting copies of itself into other DNA sites within the same cell. So that's what they call it, a parasitic DNA. Transposons. And then what this one is saying, the piwi-associated RNA induced heterochromatin block the expression of these parasitic RNAs. Okay. RNA-based regulations of chromatin structure is likely to play an important role in the gene regulations. siRNA may have evolved first, followed by miRNA later on, and later piRNA. A program of different differential gene expression leads to the different cell types in multicellular organisms. Okay, now we're talking about at the beginning, the zygote, you remember it was one cell and become 2 cells, 4 cells, and all of a sudden, after 9 months, of course, you have the cells, like your eye cells are totally different. The shape of your rods and cones of your eye is different than the shape of your liver cells. The shape of your liver cells, and of course the function of it, different than your skin cells. The skin cells are different shape than your muscle cells. So that's called differentiation. We all started with one cell, zygote. It was just one cell, and 2 cells and 4 cells, and then 8 cells, 16, 32, and 62, 64, 32, 64, and so on and so forth. Their cells are pretty much same, but pretty soon, a few months later, 9 months later, the cells are different shape, okay? So that's what we are getting into. That's next. During embryonic development, a fertilized egg gives rise to many different types of cell types. They say there are, how many? There are 200 different types of cells in our body. Cell types are recognized successively into the tissues, organs, systems, and the whole organ system. Gene expression orchestrate the developmental programs of the animal. So a genetic program for embryonic development is a transformation from zygote to adult, results from cell division, mitosis, cell differentiations, and morphogenesis, shape of change, and stem cells is a cell differentiation. Here's a fertilized egg eventually becoming a new tadpole. Cell differentiation is the process by which cell becomes specialized in structure and function. I already talked about that. The physical process that gives give an organism its shape constitutes of morphogenesis. Okay. Differential gene expression results from gene being regulated. Of course, gene regulation is involved here heavily in each cell types. Materials in the egg sets up regulations that is carried out as the cell. So what happens here, you have, for example, in frog, of course, I did not draw the nucleus here for you. I did not draw the nucleus for you here. If the frog cells you have, at this end of the cell, you have A, B, C, D molecule. In a human, this is the nucleus. You have X, Y, Z molecule right here. That's what makes a frog cell and a human cell different, because of these protein molecules in the egg. Those protein molecules make a frog, the expression of the genes. The DNA is, I didn't say the gene is same in the frog and human, but DNA is same is in frog and in human. Same DNA, A to T, G to C. You remember that? Same. But the protein molecules that are in the egg makes it different. We become human, and the protein molecules in the frog is different. So it makes it a frog. What genes are expressed? Cytoplasmic determinant and inductive signals. An egg's cytoplasm contains RNA, protein, and other substances that are distributed unevenly in unfertilized egg. I talked about that a little bit in the last diagram. Cytoplasmic determinants are maternal substances in the egg that influences early development. As the zygote divides by mitosis, cell contains different cytoplasmic determinants, which leads to different gene expression. I think these are all elaborative of what I said so far. For example, temperature can make a difference in what animal becomes. It's just not a molecules inside of the egg, it's the environment can play a factor too. In case of turtle, at low temperature, the animal becomes male. In case of alligators and crocodile, low temperature makes a female. High temperature, opposite of that, makes a female for turtles, and a high temperature for alligators and crocodile makes a male. So the other important source of developmental information is the environment around the cell, especially signals from the nearby embryonic cells. Incubation temperature determines the sex of the cell. In the process called induction, signal molecules from embryonic cells cause transcriptional changes in nearby target cells, and I will talk about that with diagram. The interaction between cells induce differentiation and specialization right here. So if you have, unfertilized egg and sperm becomes fertilized egg, these are the protein molecules that I was talking about right here. So what happens, one cell becomes two cells become four cells, but these cells release it's called induction, release the molecules, and these molecules in the adjacent cell next to it, makes it a cell of brain, a neuron. Makes it a cell of this one, it makes a cell of muscle. And that's called induction. Differentiation is taking place, but induction is a sub portion of differentiation, if you would. So this molecule makes brain cells, the triangular, green triangular, the round brown makes muscle cells. Of course, they're released from these stem cells. They're released from the stem cells, and the next cell they go to, it triggers the machinery, the genes of the next cell, and that cell becomes muscle cells. Cytoplasmic determinants in the egg, right here, molecules of two different cytoplasmic determinants. The nucleus, of course, fertilization takes place, and two cells embryo, and right here is induction by nearby cells. It's released to the next cell, and of course, they have the receptors. What is this? Oh, come on.
Cell-to-cell signaling with molecules carrying signals from sending cells to receiving or target cells.
Oh. Let's start all over.
Cell-to-cell signaling with molecules carrying signals from sending cells to receiving or target cells is a key mechanism in the coordination of cellular activities. In most cases, a signal molecule acts by binding to a receptor protein in the plasma membrane of the target cell and activating a series of changes, a signal transduction pathway, that triggers a specific response within the cell. A signal molecule binds to a specific receptor in the plasma membrane of the target cell. This activates a series of relay proteins, which in turn activates a transcription factor. The activated transcription factor attaches to the DNA and triggers transcription of a specific gene. The mRNA message is translated into a new protein, which changes the structure or function of the receiving cell.
Okay, so we're talking about induction. And then sequential regulation of gene expression during cellular differentiation determination irreversibly commits a cell to its final fate. Determination precedes differentiation, and cell differentiation is marked by the production of specific tissue-specific proteins. Okay, myoblasts are cells determined by produced muscle cells and begin producing muscle-specific proteins, and MYOD is a master regulatory gene, encodes a transcription factor that commits the cell to becoming skeletal muscles, and MYOD proteins can turn some kind of differentiated cells, fat cells and liver cells into muscle cells. Here they are. Again, I'm not going to talk about it, these in great detail. Patterns formation, setting up the body plan. Pattern formation, again, that is important in animal. Pattern and positional information, the molecular cues that control pattern formation, like anterior, posterior cells. Drosophila, which is a fruit fly. It's great to study for genetic phenomena. So determines the unfertilized egg, determines the axis before fertilization. After fertilization, the embryo develops into segments larvae right here. Right. So here you have an egg, and we talked about this nurse cells. So the nurse cells go to that one end, and the other cell, the egg is here, and eventually this becomes the anterior of the animal, again, because of these depleted nerve cells, and then and this becomes the posterior of the animal, which you have the anterior here and posterior of the animal right there. Genetics, always analysis of early development, scientific inquiry. Lewis and Noslin, they won the Nobel Prize, decoding pattern formation of Drosophila, discovered the very good homeotic genes, which are controlled, very important, you should know this, control patterns formation in late embryo, larva, and adult stages. Homeotic genes are genes that tell where the body plan goes, such as leg, arms, and head. If you want to mutate an animal, you make the egg, you saw it earlier in semester, you want to make from the eye, egg comes out in fruit fly. Well, you have to fiddle with that gene. You fiddle with that gene, you can make an egg comes out of the eye, I guess right here. As you can see, this is the wild type normal. This is the eye. And here in the eye, you have the eggs coming out because they played, these scientists were played, which they end up winning the Nobel Prize. They played with a homeotic gene. Access establishment, maternal effects genes encodes cytoplasmic determinants that initially establish the axis of the body of the Drosophila. These maternal effects genes are also called egg polarity genes because they control the orientation of the eggs. Okay, that is, the egg polarity is important when you get polarity. When you get to zoology, we'll talk about that. That is something for you guys, you should know. Okay, one maternal effect gene, the bicoid genes affects, there are a few quiz questions, exam questions from this. The front of the body of the plant, an embryo whose mother has no functional bicoid genes lacks the front half of its body. So bicoid gene, remember I was talking about posterior and anterior. These are the genes determine the posterior and anterior of the animal. They spelled out posterior for you, but they didn't mention. Here they are, bicoid morphogen that determines the head. You have the head, you have the tail, and then if it is mutated, there is a tail, and there is a tail, there is no head. Okay, the other one, high bicoid region, the results, fertilization, translation of bicoid mRNA, and then a low bicoid gene, and then bicoid proteins in embryo, and showing you what is happening.
Over the last few years, research has shown that mRNA molecules in the unfertilized egg of Drosophila determine the embryonic axes and future body plan of the fruit fly. The mRNA molecule that specifies the anterior-posterior head-tail axis of the embryo is transcribed from an egg polarity gene, which codes for a protein called bicoid. This gene is active, mRNA is made, in the nurse cells that surround the developing Drosophila egg. The bicoid mRNA, once transcribed, is deposited into the Drosophila egg asymmetrically. More bicoid mRNA is deposited at one end of the egg than the other.
Very good.
Fertilization of the egg triggers the translation of the maternal bicoid mRNA into protein. The bicoid protein then diffuses through the egg, forming a gradient. The bicoid protein gradient establishes the polarity of the embryo along the anterior-posterior axis. The head and thorax will develop in the zones of high bicoid concentration. The abdomen will develop in the region of low bicoid concentration.
Okay, that comes in handy when we go to zoology and you see the horizontal and vertical cleavage. You ask me, Amir, what is that? This piece of information you need. The bicoid research was groundbreaking for three reasons. It identified the specific protein requirements for some early steps in pattern formation. Yes, it increased understanding of the mother's role in embryo development. And then another thing about mother's rules, the mitochondria, the sperm does not have mitochondria. Well, it has mitochondria, but it does not enter the egg. So all of your mitochondria is from your mother. And that's another importance of mothers in the embryology. It demonstrates a key developmental concepts that a gradient of molecules can determine polarity and positions, of course, which we'll talk about. The next one, your textbook is getting into cancer. So it should have been separated a little bit. So these oncogenes are cancer-causing genes in some type of viruses. But proto-oncogenes are corresponding normal cellular genes in us, responsible for normal growth and cell division. We do have proto-oncogenes. Conversions of proto-oncogenes to an oncogene, the oncogene that causes cancer, we do have proto-oncogenes. it becomes oncogenes, lead to abnormal stimulation of the cell cycle, which cancer, we studied it a little bit in the past. Let's talk about it here a little bit more with gene regulations. Okay, tumor suppressor genes normally help prevent uncontrolled cell growth, okay? And then mutations that decrease protein products of tumor suppressor genes may contribute to cancer onset. Okay, so we do have these protein molecules, the genes is called tumor suppressors in our cells. Here's a gene. This segment is called tumor suppressors, and they make protein molecules that those protein molecules, it stops the oncogenes, okay? But mutation in this segment, the radiation, the food we eat, environment, stress from environment, all of those can cause mutation in this segment. And when this segment is mutated, then the protein molecules that stops the oncogenes are not made. So the proto-oncogenes become oncogenes. I hope I'm making some sense a little bit. May contribute to cancer onset. Tumor suppressor proteins, repair damaged DNA, control cell adhesions, and act in signalings that are pathways that inhibit the cell cycles. Yes, they stop cell cycles. Okay, interference with normal cell signaling pathways, mutations in RAS, proto-oncogenes, and P53. I will talk about them a little bit, both of them in a minute, hang on. Tumor suppressor genes are common in human cancers. So, RAS. Mutation in RAS can lead to production of hyperactive RAS proteins and increase cell division. So RAS is not a good gene, unlike P53, and I will talk about P53. So RAS, if it is mutated, it leads into cancer. So it's just not one gene or two genes in our body, it's a collective of genes, and then you're mentioning two of them in here. Okay, RAS is a bad gene if it is mutated. Okay, P53, if we can make more of P53 in our body, we are saved. So suppression of the cell cycle can be important in the case of damage to a cell's DNA. P53 prevent a cell from passing on mutation due to a DNA damage. Okay, so mutation in the P53 gene prevent suppression of the cell cycle. As I said, P53 is a good thing to know. Here's a diagram. I'll let you guys study it and acclimate yourself. As I said, UV light, it can cause cancer, stress from environment, and so on and so forth. The multi-step model cancer development, as I said, cancer is just multiple of things. It's just not one thing. Multiple mutations are generally needed for a full-fledged cancer. Thus, the incidence increases with age, of course. And at the DNA level, a cancer cell is usually characterized by at least one active oncogene and the mutation of several tumor suppressor genes. Right, that's a key thing. Right here. Okay, the mutation of the breast cancer, the BRCA1 and BRCA2, these are genes. We male get breast cancer too, not as often as females, but here's a mutation in BRCA1, two genes that are identified in females. At least half of the inherited breast cancers, and tests using DNA sequencing can detect these mutations. Here's a colon cancer, loss of tumor suppressant APC, very important exam question, or other genes. So APC, a suppressor gene, is found in colon, normal colon cancer, it's showing you normal epithelial cells in the cancer in normal person. small benign growth polyp right here, and then loss of tumor suppressors gene SMAD4. Do not worry about that gene, but APC, I asked a question from it. Activation of the RAS oncogene, RAS becomes activated and larger benign, so and becomes bigger, loss of tumor suppressor, P53, and then voila, you have a huge cancer. Additional mutation, malignant tumor. So the rule of viruses in cancer, a number of tumor viruses can also cause cancer in human, and viruses can interfere with normal gene regulations in several ways, integrate into DNA. Human papillomaviruses, as you know, now recently the vaccine came for it. It can cause cervical cancer, viruses, pathological agents. Okay, so I think I'm done with this chapter talking about different things. We talked about the inputs and at the end. Yeah, this is a great, it puts everything together for you guys. So make sure you study that. I think I'm done with this chapter. And if there's any question, a very important chapter, if you guys go into biological field, make sure you know, you study this chapter. Very, very important.